Okay, we're going to talk about proteins. And proteins are polymers of amino acids, and that is one amino acid I've just drawn there. Okay, so just like starch is a polymer of glucose, proteins are polymers of these building blocks, these monomers, amino acids. Proteins are you can't overestimate the importance of proteins in biology. All enzymes are proteins. Quite a lot of hormones like insulin is a protein. Hemoglobin, which carries oxygen, that's a protein. You find them as channel proteins in cell membranes. They catalyze all the chemical reactions in a cell. They are incredibly important, and there's a huge variety of them. Okay, so amino acids, you can see that we've got the usual rules here: carbon forming 4 bonds, nitrogen forming 3, oxygen 2, hydrogen 1. And the reason why they're called amino acids is because you have this group here is the amine group, and this group here is the carboxylic acid group, which we've come across before in GCSE, ethanoic acid in vinegar. When we're talking about lipids, you've got fatty acids, they contain the carboxylic acid group. So they've got two different groups, functional groups in one molecule. They all have this hydrogen there, which doesn't do that much, but then importantly, this group R, right? There are 20 different R groups in naturally occurring amino acids. So that means there's basically 20 different amino acids, and they can be joined together in almost an infinite number of ways. You can imagine the average protein may be 400 or 500 amino acids long, and you've got 20 different amino acids. Each one can be used several times or not at all. The permutations are enormous. Just to give you an idea, well, the simplest thing that R can be is simply a hydrogen atom, and that's called glycine. It could be a carbon with 3 hydrogens attached, the CH3 group, and that is alanine. Some of them contain, and we'll talk about this later, a sulfur atom. That would be cysteine. That's pretty important. Some of them contain charge groups, and that's also quite important. So, for example, aspartic acid has a negatively charged group. Lysine, that contains a positively charged group, and that's going to be very important because you can see these two, the negative and the positive, from different parts of the protein can interact with each other. So we'll talk about that later. But now we just need to see, how do they polymerize? How do they join together? Well, it's quite a familiar sort of reaction here. It's a condensation reaction. And remember, in a condensation reaction, you make a molecule of water. Okay, so what happens is you can use H2O there. Use H2O there. I've drawn 3 different amino acids. I've drawn them in 3 different colors, so each one would have a different R group. And these can join together to form, you wouldn't really call it a protein. When it's short, you call it a peptide. So you call this a peptide, and it's 3 long, and we would call it a tripeptide. Incidentally, I think one of the shortest proteins is oxytocin, which is involved, sometimes called a love hormone. It's a hormone which is involved in feeling empathetic towards people, and it's also involved in causing contraction of the uterus during labor. It's involved in lactation following birth, and that's only about 7 or 8. I think it's 7, maybe 8 amino acids long. That's very short. Most of them are quite a lot longer than that. Okay, so there's our tripeptide, and we can see that this bit here, we call this bit, that's called a peptide link. Okay, it's similar to an ester link, not quite the same. We come across an ester in there. So there's a peptide link there. So there's 2 peptide links in the tripeptide. It's also quite important to realize that proteins, they're not the same at both ends. And this is usually called, this end here is usually called the N terminus. Terminus is the last stop on the bus, isn't it? And this is called the C terminus. So it's not symmetrical, it's got different ends. Right, So so what we've got there is I've drawn here is a string of amino acids all joined together. Okay, so I've got the red one, blue, and so on. Yeah, it's a different sequence there. Now that sequence of amino acids is called the primary structure of a protein. The primary structure, sometimes called like that, primary structure. So that's just simply the sequence of the amino acids, but it never exists just as a long straight chain like I've drawn it there, because as it's made, as one amino acid is joined onto another, and this happens on the ribosome in the rough endoplasmic reticulum, it starts folding as soon as it's made. So it folds immediately into a three-dimensional structure, and the three-dimensional structure is what gives the protein its function. So if it's an enzyme, you've probably heard of the lock and key hypothesis, where the substrate has to fit into a little pocket and the three-dimensional into the surface of the enzyme. Or if you had hemoglobin, it's got a little pocket for oxygen to fit in there. The structure of a protein, the three-dimensional structure, is all important. That's what makes it do what it does. Okay, so what is it that holds together this 3D structure? Now, first of all, we need to consider, we've got the primary structure there. We're now going to talk about the secondary structure. Secondary, or so it's like that. Okay, now I've got two things here. Don't worry about, look at the blue things, okay? So look at this blue coil. This is called an alpha helix. Now, very often, when a protein assumes its three-dimensional structure, it will have parts of it which are like a helix, twisted around like that. And these little red dots I've drawn there, they're called hydrogen bonds, which I haven't mentioned before. But hydrogen bonds, these are intermolecular forces. They're not covalent bonds. The IM forces, they're not covalent bonds. So they're a lot weaker than covalent bonds. The name is a bit confusing because it says hydrogen bonds. Now, a hydrogen bond is what holds the alpha helix together. Now, what is a hydrogen bond? Well, there's an attraction because this hydrogen here on the nitrogen is a little tiny bit positively charged, and the oxygen here on this carbon-oxygen double bond is a little bit negatively charged, and they attract each other. It's quite a strong, well, nothing's like as strong as a covalent bond, but there's some. So as intermolecular forces go, it's a reasonably strong attraction. That's what's holding this alpha helix together. Now, I say hydrogen bonds are reasonably strong, but if you increase the temperature, the whole thing's going to start vibrating. You get to a certain temperature, the vibrations are so big, the hydrogen bonds are not strong enough to hold the shape, and that will break down. And that temperature is at about 37 degrees C, which may be familiar to you because you know that temperatures above 37 degrees C, proteins are said to be denatured, and that's why they're denatured at that temperature, because the hydrogen bonds break, so it loses the three-dimensional structure, and so they don't work anymore. So if it's an enzyme, the function of the enzyme is dependent on the 3D shape. Because it hasn't got that shape anymore, it's not going to work. Right, alpha helix there, also held together. Sometimes you get this in proteins. Slightly less common is the beta sheet, or sometimes it's called the beta pleated sheet. Okay, and here, rather than the peptide chain coiling, it goes like a snake, yeah, like that. But again, what's holding it together are, the little red dots there is hydrogen bonds. Okay, so again, it's the attraction between the hydrogen on the nitrogen atom and the oxygen on an oxygen on another amino acids. Incidentally, the alpha helix, these hydrogen bonds they're from the nitrogen on one amino acid residue in the protein, and the carbonyl group is called the oxygen, 4 amino acids down in the chain. So every 4 amino acids is a hydrogen bond. Right, now that's not the only level of of arrangement in the protein. So the primary structure, simply the amino acid sequence. Secondary, this is the alpha helix and the beta sheet. But we can also get a tertiary structure. So what I've got here, this is, we have a look here. So that's taken from the Wikipedia page, and the acknowledgement is just down below there. All right. So right. So I'm talking about tertiary structure. Okay. So you can probably see there the gold sections. They are regions of alpha helix. So you can see you get regions of alpha helix, and the helix breaks. Like for example, see there, that's just a grey line. There's no alpha helix there at all. So you get one alpha helix like you've got running this way, you've got one running that way, and altogether these combine to make a three-dimensional structure of the protein. This is actually amylase from a barley. So what amylase does, it breaks down starch. It's an enzyme. Breaks down starch into maltose. It's present in barley seedlings. You can probably also see here these blue bits. Can you see them there? That is a small region of beta pleated sheet. So some proteins contain regions of alpha helix and beta sheet. Some just all alpha helix. I think for the silk protein of the silkworm makes, that's mostly beta sheet. Different proteins have different amounts of alpha and beta in there. And the way those are arranged, well, that is called the tertiary structure. Okay, now what's holding the tertiary structure together? What is it? Well, there's various things. Okay, now there's an amino acid called proline. Okay, the R group is different, and it can't form H bonds. So very often when you stop like there, you might have a proline there, okay? Because that's where the helix stops. You might have a proline there, okay? It's called the helix breaker sometimes, okay? So that will affect it. Also, I mentioned earlier, some R groups like the cysteine, it contains the R group. It's actually a CH2 and an SH. Now, what happens with cysteine is it can form disulfide bridges. Okay, write that down. So, what I've got here is I've got a bit of polypeptide, a bit of protein, and I have a cysteine residue sticking out here, SH, and another one here, SH. Right, and what will happen is, well, after the protein's been made, an enzyme comes along and removes those hydrogens. It removes those hydrogens, and it forms a new covalent bond between the cysteine, between the two cysteine residues. So that is a covalent bond. Now, covalent bonds are strong. They're not like hydrogen bonds that are broken at 37. If you boil a protein up, you're not going to break that cysteine, that disulfide bridge, but it is important in holding one part of the chain next to another part of the chain. So that's what will cause the protein, pretty important, is overall three-dimensional shape. Okay, as well as cysteine residues, earlier on I said that some of the amino acids can have charged residues. I'll give you the names and you look them up if you're interested, but aspartic acid is one amino acid, and that has got a negatively charged, I'll write it like that, COO minus. Another amino acid, lysine, that has got a positively charged nitrogen in it. Right, so if you get a section of peptide with aspartic acid here, COO minus, and you get another one, so here you've got a lysine, which is H3N plus. There's obviously an attraction here between the positive and the negative. That's called an ionic interaction to ions, and that's going to hold it in place too. It's going to, right, the ionic reactions are very sensitive to changes in pH. And you know, if you change pH, usually you end up denaturing a protein. You make it very acidic or very alkaline. And the reason why is because you'll change the charges here. Okay, messing about with pH will get rid of some of those charges. If you make it too acidic, you'll get rid of the negative charge. Too alkaline, you'll get rid of the positive charge. You'll no longer have the opposite charges attracting. You'll break, the protein won't be held in that 3D shape. So the tertiary structure, yeah, is right how these regions of alpha helix and beta sheet are held together in a bigger, in a larger arrangement. Right, now finally we need to talk about quaternary structure. Okay, quaternary, do that. Well, right, now very often proteins are not just made up of one polypeptide chain. They're made up of several polypeptide chains which are joined together either by these intermolecular forces or by covalently, but they're associated together. Here, this molecule here is hemoglobin, the oxygen carrying protein, and there is the, from the Wikipedia page, and there's the acknowledgement, someone called Zephyrus, who does a lot of beautiful diagrams like that. So there's hemoglobin. Now you can see there, you've got red and you've got blue. Now the red are called, the red is the two red bits of two polypeptide chains, they're identical, and they're called the alpha chains. Don't confuse that with the alpha helix, they just call them alpha because it distinguishes them. And the blue, they are called the beta chains, and you've got two of them, they're identical to each other. And the quaternary structure is how they're arranged together. You can see there's loads of regions of alpha helix in that protein there. I can't see any beta sheet regions, but there's tons of alpha helix and doubling back on each other, giving it this overall structure. You can probably also see these green bits. Can you see there? Okay. Now a lot of protein. This is a non-protein bit, and it's a lot of proteins have these, and in this case, it's called the heme group. It's a fairly small organic molecule, but it contains an iron atom. Okay, an Fe2 plus ion, well, an atom actually, and this is the oxygen binds to that. That's what the oxygen actually binds to. You can see you've got 4 heme groups there, and they're covalently bonded, one on each alpha chain, one on each beta chain, and that whole arrangement of the 4 different chains and the 4 heme groups, the non-proteinaceous things, that is what's referred to as the quaternary structure of the protein. Okay, so there you have it. Primary, secondary, tertiary, and quaternary levels of organization, all giving each protein its own unique shape, which is absolutely crucial to its function. If you do anything to change that shape, the protein is denatured, doesn't work anymore.